Welcome to AMiNDR, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful. Hello. How are you? I know I can't hear you, and I'm not gonna lie. It feels odd to be speaking in the void, but I hope that one day I will hear back from one of you by email or Twitter or Facebook or Instagram. We're everywhere. Just find us. Welcome back for another episode on amyloid beta and Alzheimer's disease. This is our last stretch for the June papers. Woohoo! Only a couple of episodes left before we move on to the next month. We are skipping July, and we're currently working on the papers published in August 2020. Our team is working super hard. Really, we're on this every day. So if you like our episodes, please subscribe on whichever channel you get your podcast from, and let us know how we're doing. This is Sarah, hi, and I'm hosting the second stretch of abstracts addressing amyloid beta pathology in AD. I hosted another one on APP processing and amyloid beta aggregation, which I think you'll find useful if this is what you study. I have my friend and former colleague from the Cashman lab, Shyu, to thank for summarizing these abstracts. Thanks, Clay. In case you're new to our podcast, I'll let you know that we do not select papers for their quality or their merit. We cover all the primary literature, which we sort into themes, so one theme per episode. And we keep to summarizing the abstracts, so if something seems interesting to you, make sure you check the original source. We generate the timestamped bibliography to make this easier for you. You can find all the details on how to access this bibliography in the episode notes. Or just tweet at us if that's easier. Okay, let's get down to business. We have 10 papers today, and the first one is kind of a standalone. Linking cognition to age and amyloid beta burden in the brain of non-human primate, Microcebus murinus, or in other words, the grey house lemur. First author is Schmidtke, and last author is Maître Français. Okay, it was a collaboration between a lab in Germany and one in France, so I hope I pronounced them correctly. I apologize if I didn't. And you'll find this paper published in Neurobiology of Aging. Okay, here the authors use memory and learning tests along with an immunological assay of the brain in the grey mouse lemur, or Microcebus murinus. They wanted to establish a correlation between protein pathology and cognitive decline due to aging. The analysis shows that age positively correlates with worsening cognition. Now, on to the part that is the focus of this episode. A subsequent examination links amyloid beta and tau deposition to the observed performance decline with age. This represents the first study to directly establish this relationship in a non-human primate, according to the abstract. That was the only paper in the series examining a link between amyloid beta burden and cognitive decline. Now, let's take a closer look at pathological changes, starting with structure-toxicity of amyloid beta aggregates. You'll find that the first paper, the next cluster, is a bit of a Swiss knife. It touches on amyloid beta aggregation, toxicity, but also potential therapeutics for AD and type 2 diabetes mellitus. Its title is Amylin and beta amyloid proteins interact to form amorphous heterocomplexes with enhanced toxicity in neuronal cells. You'll find it published in Scientific Reports by Baradouaj, first author, and last author is Newsholm. This group looked at the potential interaction between amyloid beta and human pancreatic islet amyloid polypeptide. I'll refer to that as HIAPP for simplicity. They found that HIAPP increases the formation of A-beta oligomers as well as small and large heteroaggregates. Okay, I wonder what the criterion was to distinguish oligomers from other aggregates. These mixed complexes present unique and diverse structures and lead to greater cell death when compared to A-beta and HIAPP alone. Interesting. So these complexes were more toxic than A-beta alone. The presence of a similar but non-amyloid generating IAPP, however, reduces A-beta cell toxicity. And this similar IAPP was a rat amylin, so RAAPP. This was independent of its ability to complex with A-beta. These results suggest further study into IAPP A-beta complexes and potential for IAPP as therapeutics in Alzheimer's disease and type 2 diabetes. This reminds me, there are some papers we touched on in our episode on repurposing drugs from February and June. If you're interested in diabetes and AD, you should check those out. Now for our next paper, prepare yourself to hear about antibodies, amyloid-beta oligomers, toxicity. Okay, why do I even try? The title does a better job than me in summarizing this. Wait for it. Rationally designed antibodies as research tools to study the structure-toxicity relationship of amyloid-beta oligomers. Neat, right? The first author is Lim Brocker and last author is Vendrus Kolo, and you'll find this paper published in International Journal of Molecular Sciences. If you work with amyloid-beta, you would know how difficult it is to target the very short-lived A-beta oligomers. The authors in this paper wanted to better characterize the toxicity and structure of these oligomers. Don't we all? Don't we all? Okay, their approach is quite similar to what our lab does, so consider me intrigued. Here's what they did. They used two rationally designed single-domain antibodies. One targeted residue 18 to 24, and the other one targeted residue 34 to 40. And all of this was in A-beta 40, not A-beta 42. The application of these antibodies to zinc-stabilized oligomers led to an increase in size and hydrophobic exposure. Simultaneous increase in both parameters canceled out the individual effects on toxicity. And this is in line with current understanding of structure-toxicity relationship. This demonstrated that single-domain antibodies could be used as tools for A-beta oligomer examination. Still on the topic of amyloid-beta-mediated toxicity in neurons, the next paper is titled Roles and Interaction of the MAPK Signaling Cascade in A-beta 25-35 Induced Neurotoxicity using an isolated primary hippocampal cell culture system. Whew, this is all in one breath. Okay, so A-beta 25-35, that's what they looked at, not A-beta 40, not A-beta 42. First author is Ilun, last author is Ahmadiani. And you'll find this paper published in Cellular and Molecular Neurobiology. The authors sought to better understand how A-beta disrupts mitogen-activated protein kinase's function, or MAPK function. To do this, they treated primary hippocampal cells with A-beta 25-35 and with MAPK inhibitors. Okay, we're about to get real technical here, so get ready. If you have a pen and paper, perhaps drawing this will help out. MTT assay revealed A-beta increased cell death in a dose-dependent manner. There's more. This was exacerbated by JNK inhibition, but not by ERK or P38. Western blots showed that A-beta increased JNK, ERK, and the phosphorylation of CGUN, or CJUN, but not P38. Inhibition of P38 decreased ERK phosphorylation, and JNK inhibition lowered CJUN expression. All of which illustrated the complexity and interdependence of MAPKs in A-beta toxicity within the hippocampus. I told you this was going to be technical. Sorry, not the best for a podcast format, but hey, if any of those pathways was relevant to your work, check our timestamp bibliography to find the original paper. Details to access it in our episode notes. I know this is a short episode, but I really like the track that Anusha created and that we usually use for our breaks, so enjoy a few seconds of it. If you listened to the first part of this episode on amyloid beta, you may remember the multiple papers on membrane structure and membrane integrity in the cell. Well, our next paper looks at amyloid beta-mediated damage of the cell membrane. You'll find it under the title, A-beta 1-42 tetramer and octamer structures reveal edge conductivity pores as a mechanism for membrane damage. First author is Ciudad, last author is Carola, and you'll find it published in Nature Communications. In this paper, the authors use molecular simulation to model the behavior of A-beta with lipid membrane. A-beta 1-42 form tetramers, and now get ready for description. These tetramers had a hydrophobic center region flanked by hydrophilic residues, which allowed for membrane insertion. Increasing A-beta 1-42 concentration produced octamers, which were assembled from 2 tetramers. Membrane insertion resulted in pore formation and disruption by allowing water to go through. Ouch. Bye-bye cell. If you want to hear more about amyloid beta aggregation and cell membranes, I hope you enjoyed the first part of this episode, really. We released it on August 18th. It's a little longer than this one, but oh boy, there are a lot there on APP processing and oligomer formation, and different types of A-beta aggregates too. Okay, our next paper looks at different pathway with a focus on annexin 5. It was published in Scientific Reports by first author Bartolome, last author Scaro, and the title is Annexin A5 prevents amyloid beta-induced toxicity in choroid plexus. Implications for Alzheimer's disease. Based on a previous study looking at protein level changes in AD choroid plexus, this group focused on annexin A5. In case this is new to you, the choroid plexus is a structure forming the blood-cerebrospinal fluid barrier. As for annexin 5, it was a protein shown to prevent A-beta toxicity. Examination of AD in mild cognitively impaired patients revealed that as disease and A-beta deposition worsened, there was a reduction of annexin A5 in the choroid plexus. However, it increased in the CSF. These changes were reflected in vitro. When A-beta was applied to rat primary cultures of choroid plexus cells, there were time-dependent changes in the levels of annexin A5. Now, what happens when you try to rescue this with adding annexin A5 to the primary culture? Well, this intervention alleviated the autophagy impairment and apoptosis that were caused by nasty A-beta. This suggests a protective role for annexin A5 in the choroid plexus, and this role gets overwhelmed with increasing A-beta deposition as disease worsens. In the other episode I hosted on this topic, I came across a paper that described A-beta toxicity as A-beta intoxication. It was so funny. Like, why am I not using those words here? Our next paper also looks at the effects of A-beta intoxication, and it's titled New Mechanism of Nerve Injury in Alzheimer's Disease, Beta-amyloid-Induced Neuronal Pyroptosis. It's published by first author Han, last author Jiao, in the Journal of Cellular and Molecular Medicine. This group wanted to understand how A-beta 1-42 may cause neuronal pyroptosis. Birks-Bacon and colleagues describe pyroptosis as a caspase-1-dependent cell death that is inherently inflammatory. For your information and for mine by the same occasion. In mice cortical neurons, A-beta 1-42 induced an increase in cell permeability and LDH release. This was the same effect observed by the positive control LPS nigerine. LPS nigerine. Okay, I tried them all. One of them has to be right? Both conditions led to increased GSDMD and NRRP proteins, which are involved in apoptosis and inflammasome, respectively. So GSDMD will be involved in apoptosis and is also known as Gesdermin D, by the way. To rescue this effect, they used nicosulfonamide, or NSA, which is an inhibitor of GSDMD complexing, and siRNA targeting GSDMD, as well as downstream caspase-1 mRNA translation. And lo and behold, yes, the increase in cell permeability and LDH release were attenuated with the inhibitor, NSA. Transgenic Alzheimer's disease mouse model injected with caspase-1 siRNA showed behavioral improvement and decreased pyroptosis in the cortex and the hippocampus. The authors concluded that A-beta induces GSDMD-dependent pyroptosis, presenting a possible therapeutic target. Alright, zooming out a tiny bit, but still looking at caspases. We'll now look at synaptic transmission and neurotransmitters, and how they're disrupted by intoxication with amyloid beta. I know, I'm having too much fun with this. Our next paper here is titled Caspase Activation and Caspase-Mediated Cleavage of APP is Associated with Amyloid-beta Protein-Induced Synapse Loss in Alzheimer's Disease. You'll find it published in Cell Reports by Park and last authors Koch. This group was interested in how A-beta induces synaptic dysfunction, and specifically, they look at caspase-mediated A-beta precursor protein cleavage, which produces toxic C31 peptide. So C31 peptide made from APP cleavage. They generated transgenic mice with APP mutation at the caspase cleavage site D66-4 or D66-4A. From these mice, they generated organotypic slice cultures which closely reproduced the cell mixtures observed in the brain. These showed attenuated synaptic disruptions and dendritic spine loss. This effect was recapitulated in control cultures treated with caspase inhibitors. Furthermore, caspase-3 activation, which is associated with spine loss, was absent in these cultures. And this supports a link between APP caspase processing and A-beta toxicity. Moving along with the synapse, let's take a look at the glutamate network with a paper titled Amyloid-beta-1-42 induced glutamatergic receptor and transporter expression changes in the mouse hippocampus. You'll find it published in Journal of Neurochemistry by Young, first author, and last author is Kwiatkowski. This group recorded the overall region and cell-specific anatomical changes of the glutamate neuronal network. They will let you know that this network is involved in AD dysfunction in the mouse hippocampus due to A-beta-1-42 intoxication. I swear, I'm going to keep using this, A-beta intoxication. Using confocal microscopy, they report a significant density decrease in the glutamate receptor subunit GLUA1 and vesicular glutamate transporter VGLUT1. Specifically, they observed this in the CA1 region of the hippocampus 30 days post A-beta injection. Dendrogyroid dorsal stratum molecularly also showed decreased GLUA1 density. Along with CA1 changes, this matched reported human AD hippocampal changes. On the other hand, other glutamate receptor subunits and transports remained unchanged. We are talking about GLUA2, GLUN1, GLUN2A, VGLUT2. Check the paper for more details. These results confirm the vulnerability of the system in Alzheimer's disease. Okay, for the last paper in this episode, let's look at a different system, shifting our focus to the cholinergic system. Ta-da! This paper is titled Amyloid-beta-1-42 downregulates adenosine 2B receptor in addition to mitochondrial impairment and cholinergic dysfunction in memory-sensitive mouse brain regions. Neat summary in the title. It was published by Samuel, first author, and the last author is Garabadu. Actually, it's a two-author paper. You'll find it in Journal of Receptors and Signal Transduction. This is oddly specific. This duo looked at the link between adenosinergic receptors to AD pathology. They injected A-beta directly into mouse brain ventricles and examined A2B receptor levels in certain brain regions. Treated mice displayed A-beta dependent decline in spatial memory performance, and this was tested by Morris water maze and YMAs 14 and 18 days post-injection. AD-like disruption to the cholinergic system was observed in the hippocampus, prefrontal cortex, and amygdala of these mice, along with a brain-wide decrease in metabolism. So look, it's quite a few things here. A2B receptor level decreased substantially in the studied brain regions of these mice, pointing to its potential as a therapeutic target. Okay, that was it. You made it till the end. I hope you come back for more. We cover lots of topics pertaining to AD, and we have episodes on brain imaging, and we have more on fluid biomarkers left for the month of June. As for our next series, we will be presenting all the themes again. Isn't that great? Until then, stay safe and keep doing awesome research. That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.